So what we're going to discuss right now is something called cell theory. I'll define that, both what cell theory is and then what theory is in particular, because that's even more important potentially at this point in time. And then we're going to look at cells for a minute, just an introduction to prokaryotic and eukaryotic. It's one of the most common things that people often bring up. What's the difference between the two? And it's one of the more important things to understand, or there's a particular aspect of this that is very important to understand that I feel most people miss. All right? And that's the surface to volume ratio and where that comes from and what it really means to the cells and how it affects them. So that's what we're going to kind of go over right now. So we're talking about cell theory. This is what we're looking at. And cell theory states there are really three kind of parts of this that cells are the smallest unit of life. All right, so that's the first one. It's not anything to really argue about. So cells are single-celled organisms. That's what we're talking about with prokaryotic cells. They're all single-celled organisms. Eukaryotic cells, some of the first groups of eukaryotes are also single-celled organisms. And then we get into multicellularity, where we have then colonial and multicellular organisms to start to create very complex sorts of things. But the smallest unit is a cell. All living things are or are made up of cells. And that's kind of what I just said, but in a more specific sort of way. That everything that's alive either is a cell or a multicellular organism that is made up of cells. Okay. So these are kind of two major parts of this, what we call cell theory. And before I really kind of get into it anymore, I want to talk about that term as well. All right. So theory. And this is an important thing to bring up because in biology we have a lot of theories, and most people do not understand what that word means. And your understanding of that word is wrong in terms of science and how scientists discuss theories. The common understanding of theory is an idea. It's like, oh, I got a theory about this, so it just means I have an idea. I sort of think this is how it might happen. I don't really know for sure. It's my theory. Okay? That's just a common colloquial way of using this word. But in science, that's not how the word is used at all. In science, we have a word for that. So theory is not just an idea, all right? An idea is called a hypothesis. So when you say, I have an idea about that, saying, I have a hypothesis is the way of saying it. So it's like, I don't know for sure if this is the reason why this happens, but I'm thinking it is, so that's where I'm going to perform an experiment to figure it out. And we'll see if the information supports my hypothesis or idea or not. Now, once you gather up enough information to really support the hypothesis, then you may accept it and use it as an explanation. At that point, it becomes a theory. A theory is an accepted explanation. Okay? It is accepted and it has data to support. So this is one of the things that is the greatest problems about maybe science in general and biology is a misunderstanding and miscommunication really between scientists and the world community, really at large. And some of it comes down to terms and the way one group of people use a particular term and another group of people use the same term in completely different ways, right? So when we talk about the theory of evolution, there are people who hear, oh, an idea, I don't know if it's really true or not. I guess not, because the scientists just call it a theory. It's like, no, they're saying the theory of evolution explains how change occurs over time. And the difference between a theory, also keep this in mind, and a law. So you might say, I've heard of laws, like say there's a physical law, well, the difference between a theory and a law generally Is it a law? And we have laws in biology too. Like when we talk in this course a little bit, and in another course that I teach with organismal biology, we have genetic laws of inheritance, so Mendelian laws of segregation or assortment. Laws in science tend to be mathematical. So often there's an equation that specifically explains that law, or that law is the equation, and you can plug numbers into it and solve and test it, and that's how it is. It's very specific and it's very defined. In biology, a theory is an accepted explanation, usually for a very large biological phenomena. It's so large it can't really be explained by just a simple equation. Not that all equations are simple, but you can't put it into just a few numbers or a few terms that can go into an equation. There's a lot more to it. In addition to that, theories are continually being tested and retested, and data is being gathered sometimes over decades or many decades that support a hypothesis, support the theory itself. So there are theories in biology that are constantly being retested and reevaluated, but when we find that we can't disprove them, we accept them, and they are the explanation that we go by. So when we talk about cell theory, it's not just an idea that all living things are made up of cells. They are, or the smallest unit of life is a cell. The last part of it is all cells come from other cells. That's, again, not a mystery or anything that we don't know is what happens. Cells divide through various types of cell division. Eukaryotes have our mitotic and karyokinesis, cytokinesis, a type of cell division. Prokaryotes have a binary fission, a particular type of cell division. Cells divide into other cells. Now, where do the first cells come from? That's a sort of different sort of question. But once you have cells, as they divide, they give rise to other cells, and then cells just keep coming from other cells from division. So these are the three sort of collective statements that we talk about when we refer to what we call cell theory. And then to kind of clear it up, theory doesn't mean just an idea, maybe cells come from other cells. No, we know this and we have information. We can watch cell division, cells will divide, and so forth. This is also where we get in the argument sometimes about a virus being alive or not alive, because a virus is generally smaller than a cell, and a virus isn't alive, and so it comes to something else. What's the definition of life, which is not what we're going into right now, but they don't meet the definition. They don't meet the criteria. They don't carry out the metabolism. They cannot self-replicate. There's a number of things a virus can't do. Just because you have genetic information doesn't mean you're alive. Just DNA in a test tube isn't alive, right? Just that alone is not life, right? Life is a whole bunch of complicated things, and that's what biology is, a fairly complicated subject, because there's a lot to it, right? There's math, there's chemistry, and there's all the things that we call biology that focus on the organisms that are alive, and the cells, because a lot of times the organism is a cell. That is the whole organism. Okay. So, putting that aside, getting into the second half of this, which is where we're focusing on prokaryotic and eukaryotic, but not everything. We're going to really have a very specific focus here on size. So, prokaryotic cells are usually referred to as being small, and eukaryotic cells are being large, question mark, or larger, probably larger than the prokaryotic cells, right? Because what does that even mean in terms of reference to? So you have to have some frame of reference. You have to have numbers to go by, right? So we go by numbers, and you go by sort of tradition, right? What you're going to find, because this is one thing people will say is, well, I just need to know some specific numbers. So we'll give you those. 0.1 to, we'll go 5 micrometers. And you're going to see sometimes it's a little U like this, M. So that is micrometer, or micrometer. That's what sometimes people pronounce it. So these are our bacterial and archaea. Right. We'll get into this a little bit more. Some of the characteristics talking about. Then we have the eukaryotic cells. Eukaryotic cells generally, again, we have to give them numbers, 10 to 100 micrometers. So you look this up. This is the kind of the generic thing you're going to find. Now, the prokaryotic cells are small. They are smaller than the eukaryotic cells. In addition to that, all right, you want to keep in mind that the eukaryotic cells, in addition to being bigger, also have more of a size range. And this is what we're going to get at with surface to volume ratio is an explanation of why, how, how do they, and why here, not only are they small, but seem to be limited in size. So they're small, and their range of size is not so great. Okay, so they're little cells, so they say the prokaryotic, and then we have the eukaryotic being bigger, but the thing is the range of sizes goes a lot bigger. And I could say, well, 100 micrometers is 10, it's just a 10 times difference. Doesn't seem like we're talking about the same thing here. And that's because this is, again, what you would generally find if you look it up in your textbook or you look up something online. Those are the numbers you'll find. You're not going to find often those things reflecting reality. Or it's more of a simplifying everything so that we can package it and give it to you in just one little piece. You could say, oh yeah, that's the case. But that's not really always the case. And there's a lot more to it to think about. So cells, when we talk about this, the size, we're usually talking about diameter. So if we have a ball, a sphere, the diameter, is the distance across it from one side to the other side. The thing is, cells aren't all the same shape, right? Some cells are longer. Some of the bacterial cells are spiral, so there's all different sorts of shapes, for the bacterial cells, right, themselves. So we can have something that is narrow, but maybe particularly long, and that is one of the things that we've now found. There's actually a prokaryotic cell, very recently sort of been found. It's called Thiomargarita. That's the genus of the organism, Magnifica. I think I've got all the letters. Yep, CA, yep, okay. So Thiomargarita magnifica is a prokaryotic cell. It's a bacterial cell, and it has been found to be 2 centimeters in length. You're saying, wait a second, that's 1,000 micrometers is 1 centimeter. So that's many, many, many times bigger than what we're saying is the average size of a eukaryotic cell. That doesn't seem to make sense. And so it is perplexing for a lot of scientists to have found it. In addition to that, one of the main things we're going to get at, it's not just that prokaryotic and eukaryotic are small. And then the other thing that we get at is what's the difference between prokaryotic and eukaryotic? Well, we focus in on this term here, karyo, which means nucleus or nuclear. Okay. EU stands for true. So when we talk about eukaryotic cells, we're talking about cells with a true nucleus. And prokaryotic cells, pro is often means maybe before. So they don't have a nucleus. Except now people have found in this organism here, this Thiomargarita, that it has a membranous structure enclosing its DNA, which is a definition that we use for a nucleus. So this is a prokaryotic cell that is technically larger than most eukaryotic cells and has what appears to be a nucleus, would be called a nucleus. So how do you explain that? Is it a prokaryotic cell? Isn't it? Maybe that makes it a eukaryotic cell. So what do we use as the definition? So the circular DNA is something that bacteria are going to have. So that's when we go into a separate talk, separate lecture, just a list of common characteristics. A challenge in biology is that you'll find there's the surface information, the basics. This is what we accept as kind of the norm for most things. If you really study biology and want to understand what's going on, and it's what makes it exciting and interesting to study biology, is you find most of the time those things, it's not that they're not true, it's just that there's all these interesting exceptions to our sort of standard way of thinking. We kind of think this is the definition, and then we find, whoa, that's not necessarily the definition because we find this other organism. So there's a lot of discovery that still happens in biology, and it changes the way we think of things, the way we define things, and the way we try to explain the history of life and how life works in general, how we even define life, because we create these terms to apply to things based on what's generally the case. Okay. Same thing goes here for eukaryotic cells. This is, we're talking about, say, regular eukaryotic cells in multicellular organisms. But what about the single-celled organisms? And in some really big, and what about giant organisms, like a giant squid? So, and that's what we kind of see here. So, giant squid. Nerve axon, okay? So axons are cells within the nervous system. We have nerve axon in a giant squid measured at about 100 centimeters. So that's, again, a single cell. 100 cells. So yeah, that's okay. Well, look, now here's the biggest sort of extreme, prokaryotic thing, which still brings us into the debate of what even is a prokaryotic cell. And now we still have eukaryotic cells that are just magnificently larger than that. Okay, so we're like, wow, what about that? And then we have other cells that are still single cells. So think about something that you're going to really kind of relate to, an unfertilized egg. An unfertilized egg is a cell. So whether we're talking about an egg of a fish, right, which can be several millimeters to like maybe a centimeter in size, amphibian eggs, also same sort of thing. They could be several millimeters, so not just micrometers, millimeters to centimeters, in size. So that's sort of, something that you could see sitting in your hand. It's that big, and that's a single cell. And then you think of eggs of larger animals, and they're technically still single-celled until the fertilization occurs and cells division starts to occur within the structure. So, it's stretching a little bit when you have the amniotic eggs and all, but you're kind of getting the idea here. That's technically what you have there. So you have some very large cells, and then you also have even a single cell of, say, a protist. So which is a green algae. I left out the R. There's a green algae called Caulerpa. That's a genus. I need to capitalize that. Okay, so prokaryotes are one thing. A protist is something else. A protist is a eukaryotic organism. Many of them are single-celled organisms. Some of them are multicellular organisms. Some of them are photosynthetic, but they're not plants. So as you start to talk about organisms, you start to get all these sort of interesting things and overlap, really between the different groups. The Caulerpa is about 15 to 30 centimeters in size, and that's a single cell. So that's a single cell, one cell, and that's eukaryotic. So, take a step back. What do we have here? So we've got prokaryotic cells tend to be smaller. Eukaryotic cells tend to be bigger. Prokaryotic cells have a traditional size range of about 0.1 to 5 micrometers. And eukaryotes have a general size range. If we kind of look at all the cells, not just focus on the one exception out of billions of different things, but most of them, 10 to 100 micrometers, still going to be much, much larger than the prokaryotic cells. However, we will find exceptions. Eukaryotic cells, there tends to be a lot more exceptions with the eukaryotes, though. This is a very, very, very extremely rare thing that's just been sort of discovered. Eukaryotes, we're familiar with all the sort of unfertilized eggs. There are protist organisms that are single-celled organisms, foraminifera and radiolarians are things. Some of them can be fairly large. So we can have eukaryotic cells that are pretty big. We know that there's other things that are outside this size range fairly commonly. So that's kind of the main thing to kind of take away here. The question is sort of one, maybe why can the eukaryotic cells get to be so big? And then what limits the prokaryotes? Why can't they get that large? So even again here, the largest one ever found in history to date, and this is really just found within the last year, still not that much bigger than the biggest, than the average, but the biggest are still way larger for eukaryotic cells. So why can eukaryotic cells just get to be so gigantic by comparison? And that's going to have to do with what we call the surface to volume ratio. And I'm going to have to just kind of end this here because I don't want to go on too long. And that's going to be where we pick up with sort of the next talk. The next lecture will be focused specifically on what we're talking about. We're talking about the surface to volume ratio. So this first little talk here is on cell theory. All living things are cells. The smallest unit of life is a cell, and so on. And then the general sizes of pro and eukaryotic cells, and some of the other sort of discussion of that. And now we're going to get into how to explain surface to volume ratio, which is something that often is asked of students in general biology or intro cell biology classes, and how can we look at that numerically and then just understand it in terms of biologically, in terms of the big picture.